Hello everyone, I am Albert Ramos and today I will be discussing with you lipid metabolism. Before we continue, at the end of this session you will be able to define lipids, describe the main types of lipids, explain how energy can be derived from fat, explain the purpose and process of ketogenesis, describe the process of ketone body oxidation, and explain the purpose and the process of lipogenesis. Lipids is an organic macrobiomolecule commonly insoluble to water but are soluble to organic solvents. These are molecules that yield high energy and have a chemical composition mainly of carbon, hydrogen, and oxygen. Lipids are important fats that serve different roles in the human body. They serve as structural components of cell membrane, function as energy storehouses, and function as important signaling molecules. Lipids has four basic groups. These are triglycerides or triacylglycerols, waxes, steroids, and phospholipids. However, 95% of fat we take comes from our diet in the form of triacylglycerols and commonly found in the byproducts like dried foods, vegetable oil, butter, whole milk, cheese, cream cheese, and in some meats, while on the other hand, nuts, corns, avocados, and olives are the natural source of triacylglycerol. Triacylglycerol is formed by the joining of three fatty acids to a glycerol backbone in a dehydration reaction. Wax coats certain marine birds' feathers and some plants' leaf surfaces. They keep water from sticking to the surface due to the hydrophobic nature of waxes. The waxes consist of long chains of fatty acids esterified into long-chain alcohols. Conversely, the 2% of the dietary lipids we take are made up of phospholipids. They are synthesized in the body to build a protective barrier or membrane to protect the cell of our body. However, phospholipids are not only present in the cell membrane. Phospholipids also form structures in the blood and body fluids in which fat is enclosed and transferred in the bloodstream. Although phospholipids are similar to triglycerides in containing glycerol and fatty acids, there are some significant differences. Phospholipids contain only two fatty acids attached to the glycerol, while the third glycerol binding site holds a phosphate group. Lastly, steroids are another class of lipid molecules, identifiable by their structure of four fused rings. Although they do not resemble the other lipids structurally, steroids are included in lipid category because they are also hydrophobic and insoluble in water. All steroids have four linked carbon rings and several of them, like cholesterol, also have a short tail. Cholesterol, the most common steroid, is mainly synthesized in the liver and is the precursor to many steroid hormones. These include the sex hormones testosterone and estradiol, which are secreted by the gonads, testes, and ovaries. Cholesterol also serves as the starting material for other important molecules in the body, including vitamin D and bile acids, which aid in the digestion and absorption of fats from dietary sources. It's also a key component of cell membranes, altering their fluidity and dynamics. The first step in the digestion of triacylglycerols and phospholipids begins in the mouth as lipids encounter saliva. Through chewing, the enzyme lingual lipase, along with a small amount of phospholipid as an emulsifier, initiates the process of digestion. These actions cause the fats to become more accessible to the digestive enzymes. As the food is being ingested into the intestine in the form of chyme, lipid metabolism begins. In the intestine, the chemoreceptors in the intestinal mucosa pick up the fat signals, and the intestinal cells in it release a digestive hormone known as cholecystokinin. Cholecystokinin, or CCK, stimulates the release of pancreatic lipase from the pancreas and stimulates the contraction of the gallbladder to release stored bile salts from the bile into the intestine. Bile salts perform an emulsification of fats in which the fat globules are converted into smaller fat droplets. The pancreatic lipases, together with a protein coenzyme called colipase, break down the triglycerides into monoglyceride and free fatty acids. The small compact molecules formed is called micelles. These fatty acids can be transported across the intestinal membrane. However, once they cross the membrane, they are recombined to again form triglyceride molecules. Within the intestinal cells, these triglycerides are packaged along with cholesterol molecules in phospholipid vesicles and other apolipoproteins called chylomicrons. Chylomicrons enable the lymphatic and circulatory systems to transfer fats and cholesterol inside the aqueous setting. By exocytosis, chylomicrons exit the enterocytes and enter the lymph system by lacteals in the villi of the intestine. From the lymphatic system, the chylomicrons are transported to the circulatory system. Once in the circulation, they can either go to the liver or be stored in fat cells or adipocytes that comprise adipose tissue found throughout the body. To obtain energy from fat, triglycerides must first be broken down by hydrolysis into their two principal components, fatty acids and glycerol. This process is called lipolysis. Lipolysis takes place in the cytoplasm. The resulting fatty acids are oxidized by beta oxidation into acetyl-CoA, which is used by the Krebs cycle. The glycerol that is released from triglycerides after lipolysis directly enters the glycolysis pathway as dihydroxyacetone phosphate. Because one triglyceride molecule yields three fatty acid molecules with as much as 16 or more carbons in each one, fat molecules yield more energy than carbohydrates and are an important source of energy for the human body. Triglycerides yield more than twice the energy per unit mass when compared to carbohydrates and proteins. Therefore, when glucose levels are low, triglycerides can be converted into acetyl-CoA molecules and are used to generate ATP through aerobic respiration. The breakdown of fatty acids, called fatty acid oxidation or beta oxidation, begins in the cytoplasm, where fatty acids are converted into fatty acyl-CoA molecules. This fatty acyl-CoA combines with carnitine to create a fatty acyl-carnitine molecule, which helps to transport the fatty acid across the mitochondrial membrane. Once inside the mitochondrial matrix, the fatty acylcarnitine molecule is converted back into fatty acyl-CoA and then into acetyl-CoA. The newly formed acetyl-CoA enters the Krebs cycle and is used to produce ATP in the same way as acetyl-CoA derived from pyruvate. If excessive acetyl-CoA is created from the oxidation of fatty acids and the Krebs cycle is overloaded and cannot handle it, the acetyl-CoA is diverted to create ketone bodies. These ketone bodies can serve as a fuel source if glucose levels are too low in the body. Ketones serve as fuel in times of prolonged starvation or when patients suffer from uncontrolled diabetes and cannot utilize most of the circulating glucose. In both cases, fat stores are liberated to generate energy through the Krebs cycle and will generate ketone bodies when too much acetyl-CoA accumulates. In this ketone synthesis reaction, excess acetyl-CoA is converted into hydroxymethylglutaryl-CoA or HMG-CoA. HMG-CoA is a precursor of cholesterol and is an intermediate that is subsequently converted into beta-hydroxybutyrate, the primary ketone body in the blood. Ketone body oxidation. Organs that have classically been thought to be dependent solely on glucose, such as the brain, can actually use ketones as an alternative energy source. This keeps the brain functioning when glucose is limited. When ketones are produced faster than they can be used, they can be broken down into carbon dioxide and acetone. Ketones oxidize to produce energy for the brain. Beta-hydroxybutyrate is oxidized to acetoacetate and NADH is released. An HS-CoA molecule is added to acetoacetate, forming acetoacetyl-CoA. The carbon within the acetoacetyl-CoA that is not bonded to the CoA then detaches, splitting the molecule in two. This carbon then attaches to another free HS-CoA, resulting in two acetyl-CoA molecules. These two acetyl-CoA molecules are then processed through the Krebs cycle to generate energy. When glucose levels are plentiful, the excess acetyl-CoA generated by glycolysis can be converted into fatty acids, triglycerides, cholesterol, steroids, and bile salts. This process is called lipogenesis. Lipogenesis creates lipids from the acetyl-CoA and takes place in the cytoplasm of adipocytes and hepatocytes. When you eat more glucose or carbohydrates than your body needs, your system uses acetyl-CoA to turn the excess into fat. Although there are several metabolic sources of acetyl-CoA, it is most commonly derived from glycolysis. Acetyl-CoA availability is significant because it initiates lipogenesis. Lipogenesis begins with acetyl-CoA and advances by the subsequent addition of two carbon atoms from another acetyl-CoA. This process is repeated until fatty acids are the appropriate length. Because this is a bond-creating anabolic process, ATP is consumed. However, the creation of triglycerides and lipids is an efficient way of storing the energy available in carbohydrates. Triglycerides and lipids, high-energy molecules, are stored in adipose tissue until they are needed. Although lipogenesis occurs in the cytoplasm, the necessary acetyl-CoA is created in the mitochondria and cannot be transported across the mitochondrial membrane. To solve this problem, pyruvate is converted into both oxaloacetate and acetyl-CoA. Two different enzymes are required for these conversions. Oxaloacetate forms via the action of pyruvate carboxylase, whereas the action of pyruvate dehydrogenase creates acetyl-CoA. Oxaloacetate and acetyl-CoA combine to form citrate, which can cross the mitochondrial membrane and enter the cytoplasm. In the cytoplasm, citrate is converted back into oxaloacetate and acetyl-CoA. Oxaloacetate is converted into malate and then into pyruvate. Pyruvate crosses back across the mitochondrial membrane to wait for the next cycle of lipogenesis. The acetyl-CoA is converted into malonyl-CoA that is used to synthesize fatty acids. To sum up this presentation, lipids are available to the body from three sources. They can be ingested in the diet, stored in the adipose tissue of the body, or synthesized in the liver. Fats ingested in the diet are digested in the small intestine. The triglycerides are broken down into monoglycerides and free fatty acids, then imported across the intestinal mucosa. Once across, the triglycerides are resynthesized and transported to the liver or adipose tissue. Fatty acids are oxidized through fatty acid or beta oxidation into two-carbon acetyl-CoA molecules, which can then enter the Krebs cycle to generate ATP. If excess acetyl-CoA is created and overload the capacity of the Krebs cycle, the acetyl-CoA can be used to synthesize ketone bodies. When glucose is limited, ketone bodies can be oxidized and used for fuel. Excess acetyl-CoA generated from excess glucose or carbohydrate ingestion can be used for fatty acid synthesis or lipogenesis. Acetyl-CoA is used to create lipids, triglycerides, steroid hormones, cholesterol, and bile salts. Lipolysis is the breakdown of triglycerides into glycerol and fatty acids, making them easier for the body to process. That ends my presentation, and thank you very much.